Telomerase deficiency impairs glucose metabolism and insulin secretion by Asif, Ladha, Ehab Anton, and Euder Menezes. Diabetes mellitus is the most prevalent metabolic disorder, and its metabolic hallmark, glucose intolerance, affects approximately 344 million people worldwide. Subtypes of diabetes mellitus. Type 1 diabetes is considered an autoimmune disease which results in complete loss of pancreatic beta cells. Type 2 diabetes is caused by a combination of peripheral insulin resistance and beta cell dysfunction. Recent evidence, however, suggests that type 2 diabetes is additionally characterized by impaired beta cell regeneration and reduced beta cell mass. Telomeric DNA is synthesized, elongated, and hence maintained by copying an RNA template sequence within the RNA moiety of an enzyme called telomerase.
Given the role of telomerase in maintenance and in replications of differentiated cells on the one hand, and the importance of beta cells losing for the pathogenesis of type 1 as well as type 2 diabetes on the other hand, it appears tempting to hypothesize that telomerase could act as a key of a beta cell viability and regenerations. Previously generated mice that is deficient for the telomerase RNA components gene is analyzed. These animals develop glucose and impaired insulin secretion due to impaired beta cells regeneration, while body fat content energy spending on insulin sensitivity were found to be inactive.
Body mass and body composition, including fat content, were measured by use of quantitative nuclear magnetic resonance technique. Food intake was recorded weekly and phases were collected 3 times per week. Phases samples were determined by bomb calorimeter. The black bar is the control animals and the gray bar is the TERK-deficient G4 animals. Figure A demonstrates that TERK-deficient G4 mice are smaller and have a reduced body mass. However, the reduction affects both lean masses, as you can see in figure B, and body fat, which you can see in figure C, to the same extent. We moreover demonstrate that both dietary energy uptake shown in figure D and energy excretion via phases, figure E, is reduced. However, when these later findings are normalized to body mass, no significant differences can be found for neither energy uptake nor energy excretion. Accordingly, determination of energy expenditure as quantified by indirect calorimeter did not show any differences regarding energy turnover at neither day nor night. You can see this in figure F. Taken together, these findings indicate that TERK-deficient G4 mice are smaller but have an unaltered relative body composition and energy metabolism.
Glucose metabolism in TERK-deficient G4 mice. TERK-deficient G4 animals are the gray line, and the control animals are the black line. Intolerance was performed on TERK-deficient G4 and on the control genotypes, and observed impaired glucose clearing in TERK-deficient 4 mice, as you can see in figure A. The aim was to illustrate whether impaired insulin secretion, or rather insulin resistance, may be responsible for this phenotype. Therefore, plasma insulin level during concentration of IBGTT was first quantified and observed impaired insulin secretion in response to inject glucose in TERK-deficient G4 mice. This shown in figure B. By contrast, performing IBITT and injecting insulin into mice which did not relieve significant difference in glucose disposed in response to insulin, which show in figure C. However, telomerase has been reported to affect mitochondrial functions, which we finally quantify glucose oxidation rate by injecting uniformly C13 glucose to quantify the exhalation of CO213, which was found to be identical in all genotypes, as you can see in figure D, precluding systematic mitochondrial dysfunction in TERK-deficient G4 mice at last in regard to glucose-derived oxidation capacity.
Since the phenotype appeared to be restricted to impaired insulin secretion, telomere length was next quantified, specifically in pancreatic islets, using quantitative fluorescence in situ hybridization (QFISH), shown in figure E and F. We observe a pronounced reduction of average telomere length in islets of TERK-deficient G4 animals, shown in figure G. Taken together, telomere deficiency causes shortened telomere length in pancreatic islets and impaired glucose tolerance as well as impaired insulin secretion in mice. Reduced beta cell mass in TERK-deficient G4 mice. Reduced insulin secretion is typically caused by reduced mass of the islets of Langerhans, which mainly contain beta cells. To decide whether this possibility may apply in regard to the phenotype observed, beta cell mass in repeated sections from pancreata of all 4 genotypes was determined by quantitative microscopy. First observation was smaller islets in TERK-deficient mice as shown in figure B, as control to control animals, which is shown in figure A. By quantifying insulin-positive areas within pancreatic sections of all genotypes, there was a reduction of insulin-positive areas per total pancreatic section as shown in figure C. Since TERK-deficient G4 are smaller than control genotypes, pancreatic and insulin-positive areas to body mass was normalized to exclude the possibility that reduced insulin-positive area size simply was an indicator of reduced body mass. After normalizing the areas for body mass, an observation of significant reduction of insulin-positive areas as shown in figure D. We moreover find a number of islets to be reduced in TERK-deficient G4 animals shown in figure E, which, however, did not apply after normalization for body mass shown in figure F. Insulin-positive area per individual islet of Langerhans is reduced in absolute terms, shown in figure G, as well as after normalizing for body mass shown in figure H. Altogether, suggesting that telomere deficiency causes a reduction of beta cell mass by reducing the size of individual islets, whereas islet number is not affected.
Impaired beta cell regeneration in TERK-deficient G4 mice. Next, bromodioxyridine (BRDU) was injected into mice prior to sacrifice, which enables us to determine the number of DNA replicating. A reduction of BRDU positive beta cells per islet was found. This is shown in figure A, as well as after normalization for body mass shown in figure B. Lastly, the number of BRDU positive beta cells per standardized insulin positive area was also found to be reduced, shown in figure C, as well as after normalization for body mass, which is shown in figure D.
As a conclusion, we here describe a direct causes link between impaired telomerase activity and impaired insulin secretion as well as a glucose intolerance in TERK-deficient G4 mice. These results are supported by the fact that shorted telomeres, as found instead of reducing telomerase activity, are associated with type 2 as well as possibly type 1 diabetes in humans.
Taken together, our findings indicate that reduced telomerase activity may be considered a cause of impaired glucose tolerance and hence diabetes mellitus. In full accordance with the role of this protein in multiple other cell types, telomerase is required to maintain the replicative potential of pancreatic beta cells. Consistently, lack of telomerase activity causes beta cell loss and subsequent deterioration of glucose metabolism, reflecting increasing incidence of diabetes mellitus with increasing age.